This conference will now be recorded.
Okay, those who have went for drives, mention their names and give it to your faculty, subject handling faculty. Okay, Vishal. Right. Okay. Good morning to all. I heartily welcome you for this final year and for this semester. This semester, I'm going to handle medical electronics, which is elective for you guys. And I hope you all uh, are ready for the placements. If some people are going for the drive, you just list out the names and hand out over to your faculty, C section, as well as the representative of A section. Collect the names and give it to me, okay, so that the faculty will put on duty for them. Right. So medical electronics, this is your course, medical electronics. Have you heard about medical electronics before? So this is an elective subject. In this subject, you are going to look at how the electronics plays a vital role in medical field. Okay, so this is a unit 1. Agenda of the session is prerequisite, cell, structure, types and functions. Okay, so human body is made up of cells. As you people are aware of cells, which you have studied in the 12th standard. Okay, basic unit of human body is cell. So how the cells are work and what is the structure and how the cells are convert your energy protein into some energy and how it is used for functioning of human activities. Okay, all this we are going to look at in first class, I mean in this today's session. So you people are very well aware of cells, is it not? Yes, cell is nothing but it is the lowest level of structure which capable of performing all the activities of life. Okay, without cell, no human body or no thing will be available in the universe. Okay, this is very lowest level of structure. We can only able to view the cell using your microscopes. Okay, so there are many kinds of microscopes for viewing your cell. This is very small and minute. Okay, so the first cell were observed and named by Robert Hooke in 1665 from the slice of cork. So first cell was developed by Robert Hooke in the year 1665. Okay, so imagine now we are in 2021. So in 1665 they have developed and established the thing called cell. Okay, so these are all some examples which comprises of cells. In plants also you could able to see some cells. Okay, in animals, in human beings, whatever the object you take, okay, living object you take will have cells inside it. Okay, the next, cell theory. So what is the theory behind the cell? Okay, it is proposed by Mathis and Theodore in 1839. Okay, so it is proposed that cell developed during the year 1665 and the theory was developed in the year 1839. Okay, what is the theory? What it expressed? The theory is expose all living things are made up of cells. Okay, first step is what? Every living thing is made up of cells. Okay, so these cells are a smallest working unit. We could able to see the cells using your microscopes, and next followed by that, all cells come from pre-existing cells through the cell division. Okay, there is some operations taken place which divides your main cell into number of multiple cells. Okay, so it is like a multiplication factor. Okay, multiplication factor in the sense 1 into 2, 2 into 4, 4 into 8, like that. It multiplies, enhances the number of cells present in your human body. Okay, it enhances the count. Okay, so some organisms consist of cells, single cell, which is equal to unicellular organism. Okay, others are multicellular aggregates of specialized cells. Okay, you could see from this diagram. First one is bacteria, archaea, protista, plantae, fungi, animalia. Okay, these are all some few examples of single cellular cells. Okay, single cellular is also called unicellular organism. Okay, so others are multicellular. Okay, other than these examples, we can also see some multicellular which aggregates or specialized cells. Okay, so these are all some single cells, kind of single cells, right? And next, followed by this, we have functions of cell. Okay, so we know cell is a vital part of every human being as well as animals and other living objects. Okay, living things. So how the cells are working? Cell is very minute structure, and what the functions of cells are as followed. Okay, so whether multicellular or unicellular, whether a single cell or multiple cell, all organisms must accomplish the same functions. Okay, it is common to everything, irrespective of human being, animals, plants, and everything. The functions of the particular cell is common, quite common. Okay, so what are all the functions of the cell? Number one is what? It uptake and processing of nutritions. Okay, so whatever the food content we are intaking, that food content processing of all the proteins, minerals, vitamins, carbohydrates, everything is processed in your digestive system. Okay, it is used to uptake and processing of nutritions, out of which we are observing some nutritions from the food, isn't it? So that nutritions is processed with the help of your cell. And next, followed by that, excretion of waste. Okay, so after processing all the food inside your stomach, intestine, that waste of the food content will be excreted out using some output organ, like output organs. Okay, say for example kidney or some other large intestine, which excretes the waste out of your body. Okay, so that is what done by your cell. Third one is what? This is response for environmental stimuli. Okay, so this kind of cells are used to respond how the environment, I mean external source will affect your cell. Say for example, if a person is affected by disease, okay, disease or some other uncurable thing, then these cells are used to, as a barriers to fight against the disease. Okay, if they have affected with some of the viruses or bacterial infections like that, these cells are having the tendency of increasing the immunity and fight against that virus or bacteria. Like that kind, it is used to respond to environmental stimuli, right? And the next step will be reproduction among others. These cells are important for reproductive system. Okay, so first step is what? Uptake and processing of nutrients, excretion of waste, response to environmental stimuli, and reproduction among others. Okay, four basic steps which is done in the functions of cell. Okay, and next followed by that, we are having how we study cells. Okay, we know what is cell, the definition of cell, and the functions of cell. Okay, so how we are going to study the cell? Okay, so we have two basic classification. One is what? Microscope, another one is self fractionation. Okay, so from the image you can see here is your size of thing will be explained in this y-axis. Okay, say for example here we have 0.1 nanometer, 1 nanometer, 10 nanometer, 100 nanometer, like this it goes up to meters. Okay, so meter, centimeter, millimeter, micrometer, nanometer. Okay, this is your hierarchy. Okay, meter it goes to centimeter, millimeter, micrometer, and nanometer. Okay, so your human height will be measured with the help of meters. Okay, and come followed by that, length of some nerve and muscle cells are measured in some meters. Okay, this meters of muscles, cells, and this meters of nerves are present in this human body, like that. And next, 0.1 m is what example we have chicken egg, and we have a few examples like plant and animal cells, nucleus, most bacteria, mitochondria, everything falls under 100 micrometer to 1 micrometer. Okay, and these all animal cell and plant cell, bacteria, everything will be viewed using your light microscope. Okay, but when it comes to this size, we can able to see in bare eyes. Okay, so unaided eye, we can also view with our eyes without using any microscopes, because the size of the things are larger when compared to this cells. Okay, when it goes down, some bacteria, viruses, ribosomes, proteins, lipids, small molecules, atoms, everything will be viewed using your electron microscope, because this is very smaller than the plant and animal cells. Okay, it is in the size of nanometers, not in micrometers, nanometers. Okay, so atoms are in size of zero point 0.1 nanometer. Okay, so most cells are between 1 to 100 micrometer in diameter, which can be visualized by light microscope. So depending upon the size of the cell and the object, we are going to visualize it. Okay, if it is under centimeters and millimeters, we can able to view in bare eyes. If it is smaller than that, it is in the micrometer or nanometer, we supposed to have some microscope to view the object and cells. Okay, so this is how we are going to study our cells, and the comparison of light microscope and electron microscope. Okay, microscope is a device which we are going to look are small size of objects. Okay, so this is of having two classification. One is what light microscope, another one is electron microscope. In under light microscope, we are using some light rays. Okay, so we are going to pass the light rays to the specimen and through the glass lenses. So I'm going to place the cell in the glass places, and I'm going to focus, I'm going to place a light over the specimen. Okay, specimen is nothing but a sample. Okay, let's say for example it is like a cell, or it is like a blood, or it is like urine, whatever the sample we are going to look at, that is called specimen. The lenses refract light such that the image is magnified into the eyes or the video screen. Okay, so what happens is when we are supposed to focus the light over the specimen, that lenses refracts, not reflect, it refracts, so that the image will be magnified. Magnified is nothing but making your image into, from small image into maximum image. Okay, I can see a large image over the screen after magnification. Magnification is also called amplification. I'm boosting the signal, I'm enhancing the signal. Okay, like that kind of operation done by your magnification. Okay, that is while refracting the light from the lens. This is the process under your light microscope. And when you consider electron microscope, we are going to focus some electrons over the specimen. Instead of light, I'm going to focus a beam of electrons through the specimen or onto its surface. Okay, so theoretically, the resolution of modern EM could reach 0.1 nanometer, but practical limit is closer to 2 nanometers. So electron microscope, we are going to use, we are going to look very minute particles like atoms. Okay, so this is very helpful for analyzing and studying your cells. And followed by that, cell fractionation. Okay, do you have any doubts in this? Am I fast? Am I fast? Do you have any doubt? No. Dharnita, as usual, you're responding. Cool. Right. And next method is self-fractionation. What is our first method? Microscope. Second method is self-fractionation. So self-fractionation is nothing but it is like a chemical reactions. Okay. So I'm going to separate the organelles of the cells for functional study. So take for example, this is your sample specimen. Okay. So I'm going to put it in some pipettes. You could see this kind of pipettes in chemical laboratory or chemistry lab, right? So what happens is I'm going to put the specimen along with some chemicals. Okay, so this is the tissue cells. I'm going to homogenate. Okay, so I'm going to mix some solution along with my specimen. Okay, after mixing, I'm going to take this sample under centrifugal force. Okay, so the disrupted cells are centrifuged at different speed and duration to fractionate the components of different sizes. So I'm going to differentiate, uh, centrifuge mix பண்றேன். I'm mixing the solution and my specimen well by the help of centrifuged force. Okay, so while I'm mixing, some of your nuclear and cellular debris is under the pipette. Okay, you could able to see here, we can have some hard materials. Okay, some hard materials which deposit the bottom of the pipette. Okay, so this is what here your specimen will be taken out with some accurate values. Okay, so this pellet rich in mitochondria and chloroplast if cells are from plants. So if I'm going to take a specimen from the plant, what happens that mitochondria and chloroplast are settled down at the bottom of the pipette. Okay, so pellet rich in microsomes, piece of plasma membrane and cell internal membrane. So hard material, most of the materials or most of the things inside the cell is weightless. Okay, the weightest portion of the particular cell will be deposited at the bottom. Okay, so that I can easily count the value of that particular things. Okay, so this is what finally will get ribosomes at the bottom. Okay, after mixing my solution with my specimen, after centrifuged force, okay, I'm going to get rich in ribosomes, which is count of the ribosomes will tells us how the particular specimen functions or the features of the particular specimen. Okay, so this is how we are going to view the thing of cells. Okay, two methods, microscope, and another one is cell fractionation. Cell fractionation la we are mixing external solution to my original specimen, and I'm undergone with some centrifuged method, that is I'm mixing thoroughly by centrifuged thing, and after many centrifuged thing, my hard parts are deposited at the bottom of my pipette so that I can able to count or view the functions of the cells. Okay, so this is second kind. And next is what? Cells. So cells are basically classified into two types. One is prokaryotic, another one is eukaryote. Okay, prokaryote, eukaryote. Okay, so in prokaryote we have nucleus at the center. Okay, so it has some flagellum and cell wall, cell membrane, capsule, ribosomes. Okay, so in eukaryote we have nucleolus, membrane enclosed the nucleolus, and mitochondria, ribosomes. Okay, so what is the difference between prokaryote and eukaryote? Each and every cells are different in nature. Okay, so your prokaryote will not similar to your eukaryote, or your plant cell is not similar to your human cell. Your human cell is not similar to animal cell. Okay, each and every cell are different from each other. Okay, so this is basic classification of cell. One is eukaryote and the one is prokaryote. Okay, so what is prokaryote? The major difference between those two is the location of chromosomes. Okay, so in eukaryotic cell, chromosomes are contained in membrane enclosed organelle, the nucleus. In prokaryote, the DNA is concentrated in the nucleoid without a membrane separating it from the rest of the cell. Okay, so chromosomes matters between your eukaryote and prokaryote. Okay, so chromosomes are contained in the memory enclosed in the organelle nucleus. In the figure you could see the chromosomes are present. These are all chromosomes. Okay, so this is present in the cell, but when you go with this one, your chromosomes will not present. Okay, so this is what given in prokaryote cell. DNA is concentrated in the nucleoid without a membrane separating it from the rest of the cell. Okay, this is basic classification of eukaryotic cell and prokaryotic cell. And followed by that, we are having human cell. Okay, so in human cell, we are having rough endoplasmic reticulum, nucleoplasm, nucleolus, cell membrane, Golgi apparatus, cells, lysosomes, mitochondria, ribosomes, cytoplasm, smooth endoplasmic reticulum. Okay, these are all units of human cell. Okay, namely centrioles, Golgi apparatus, cell membrane. These are all units. Okay, what are all the things present inside a single cell of human? Okay, it is called units of cell. So lysosomes, mitochondria, ribosomes, cytoplasm, and smooth endoplasmic reticulum. This is how your human cell look on. So cell evolution. So cell evolution, it is a fundamental structural and functional unit of all living organism. Okay. So this is fundamental unit. Without cell evolution, your function of human body will never takes place. Okay. Robert Hooke established or found this cell in the year 1665, and we have Anton von Leeuwenhoek first describing a living cell. He is the person who describes the living cell first. And Robert Brownie discovered nucleus. Okay, after finding out, after describing the thing, the cell, Robert Brown discovered nucleus. Okay, we have nucleus inside the cell, which was founded by the Robert Brown. Okay, these are all the evolution of your fundamental structure, that is fundamental unit, functional unit of living organism. And next, cell and structure. Okay, so cell and structure, the cell structure comprises individual components with specific functions and essential to carry out life's process. Okay, so cell function in and it carries your life. Okay, without cell function human will not live. Okay, this carries out life of human being. Okay, this cell is also contains body's hereditary materials. Okay, so you could have heard about the heredity. Okay, what is heredity? It is followed from the ancestors. Okay, the disease also it is followed from the ancestors. Why? Because their genetics also mixed with our genetics. Okay, so their DNA which will be matched with our DNA. So if for example, if a person is doing some crime or thing, okay, how they will identify that person correctly? With the help of the DNA only, the criminals are identified by the police department or some other department. Okay, so that minute, that accurately we can able to identify the person with the help of DNAs. Okay, so that heredity is taken from the ancestors to the generation. Okay, forthcoming generation. So cell also contain that body heredity materials. Okay, so and can make copies of themselves. Say, cells have many parts, each with different functions. Okay, as I told you earlier, this is your cell, and which comprises of many parts, and each and every parts predominantly used for doing their function. Okay, so combinedly all together which expose the function of cell. Okay, so we have components like cell wall, cell membrane, cytoplasm, nucleus, and cell organelles, etc. We have many units inside your human cell. And followed by that, we have cell numbers, shape, and size. Okay, we have seen what is cell, what is the application of cell, what is the function of cell, what are the components present inside the cell. Okay, and what are the types classification of the cell, and next comes cell numbers, shape, and size. And also we have seen how we are going to study about a cell using microscope method or cell fractionation method. Okay, two methods are available. And classification of cell, prokaryote and eukaryote. Both these types are differentiate with the help of your chromosomes. Okay, and next comes cell numbers, shape, and size. Okay, in unicellular organisms, organisms with single cell, unicellular is nothing but it is a single cell. Multicellular, more cells. Okay, so under unicellular we are having single cell which capable of independent existence. It carries all functions like digestion, excretion, respiration, growth, and reproduction. Okay, so all these functions are carried by your cell, because this is a fundamental unit of human being, right? It is having responsibility of all functions. Say for example, digestion, excretion, respiration, whatever the function done by human body, that is all because of the cells. Okay, if the cell is very healthy, then your body is also looks healthy, feels healthy. If your cells are dead, what happens? Dead cells are a long way. Dead cells are converted into tumors. Okay, that is called tumors, brain tumors, some other tumors present in the body, right? That is all because of dead cells. If we have a long way, or how to say, if we have many dead cells, okay, which sustained, which is segregated, which retains at the place for long time, okay, then that cells, dead cells are converted into tumors. Okay, so in order to avoid those things, we should engage ourselves, we should make our cells active with more functions by intaking nutritious foods, okay, and doing some workouts, everything we are going to make active our body cells. Okay, and the consumption of water is also a thing which activates, which rejuvenates the cells. Okay, consumption of water. Okay, that is why it is recommendable of consuming 3 liters of water a day, or 4 liters of water a day, like that. Okay, so all functions of human body is completely based upon the functional unit of body is cell, right? So in multicellular organisms with more than one cell. Okay, so multicellular, we are having more number of cells, and in unicellular we are having a single cell. Okay, so cell and multicellular organism vary in size, shape, and depending upon the function. Next comes shapes of the cell. We are having few classifications of shapes. One is parenchyma, sclerenchyma, nerve cells, RBCs, red blood corpuscles, muscle cells. Okay, these are having individual different shapes. Okay, so in parenchyma, it is like polyhedral cells performs storage. Parenchyma is used for storing nutrients, and it is of size polyhedral. And next, sclerenchyma, it is spindle-shaped cells and provides mechanical support. Spindle-shaped cells அப்படின்னா எப்படி சொல்றது, it is like nerve fibers madri. Okay, so it is like fibers of nerves, muscle fibers madri. Okay, that is spindle shaped. Okay, next, nerve cells எடுத்துட்டோம்னா it is like long and branched cells conducting nerve impulses. Okay, so nerve impulses is nothing but nerves, the basic unit of nervous neuron. Okay, so neuron having axon and things, dendrites. Okay, so axon and dendrites are used to stimulate the nerve operation. Okay, more millions and millions of neurons are present inside our human brain. That neuron is used to trigger or stimulate your motions, body motions. Okay, so nerve cell is a long and branched cells conducting nerve impulses. That is impulses, it is like a power to the nerves. And next, followed by that, we are having RBC, that is red blood corpuscles. Okay, this is biconcave and helps in carrying oxygen. Okay, so oxygen is very important for surviving, so oxygen is carried by your RBCs. Next, muscle cells. So cylinder and spindle shaped, concerned with the movement of body parts. So this is like a cylindrical shape, and muscles are important for motions. It is also similar, it is, everything is interlinked, and it is like a depending process. Okay, so muscle cell is used for movement of parts. So once your brain nerve or neuron triggers the pulses through nerves, the muscle also supports the action. Okay, this is responsible for actions. Muscle supports actions and do work by the human body. Okay, these are all some steps. Each and every cells of human body will have different shapes. Okay, and next, that size, so varies from few microns to few centimeters. Okay, size of your cell will vary from 1 centimeter, 10 millimeter, 10 millimeter to 1000 am. Okay, so all these are in different shapes and size. Small living cell is PPLO, that is pleuropneumonia like organism. Okay, this is very minute, 0.1 micrometer. It is similar to your atom. Okay, largest living cell is egg of an ostrich, that is 170 to 180 mm in diameter, and bacteria 0.1 to 0.5 micrometer, and sclerenchyma fibers up to 60 centimeters in length. Okay, each and everything are in different shape and different size. Followed by that, we are having cell wall. Okay, what do you mean by cell wall? We know what is cell and what is cell wall. Cell wall is nothing but the outermost layer of the cell. Okay, so this protects, this acts as a barrier. Okay, found in bacterial cells, fungal cells, and plant cells. It is permeable. Permeable is nothing but a passage. Okay, it allows things to get in. Cell wall is permeable. It is allows things to get in. Made up of cellulose. Okay, so and the function is rigidity, mechanical support, and protection. Okay, so it supports mechanical action and protect your cell. This is cell wall, which is present outermost layer of the cell. And next comes cell membrane. Cell membrane is nothing but it is the layer which present below the cell wall in plant cells. Okay, outermost membrane in animal cells. Animal cells la cell membrane வெளியில இருக்கும். The plant cell la cell wall உள்ள இருக்கும். Okay, all the cell wall and cell membrane are used to protect and support the activity. Okay, this is semi-permeable. Semi-permeable, it is not passing all the things inside. It blocks something and it allows something. It is like semi-permeable. Okay, it is made up of phospholipids, proteins, carbohydrates, and cholesterols. And the function of the cell membrane are it allows outward and inward movement of molecules across it, like diffusion, osmosis, active transpose, photocytosis, pinocytosis. Okay, these are all some functions which supported by your cell membrane. Okay, so your inhalation, exhalation, okay, oxygen infusion, diffusion, active transport of your nutrition from one part or one function to another function, all these are done with the help of this cell membrane. Okay, and next comes protoplasm and cytoplasm. So protoplasm is nothing but it is like physical basis of life. Okay, it includes organic and inorganic molecules. Protoplasm is important thing. It is physical basis of life. And cytoplasm, it is like semi-fluid matrix. Okay, cytoplasm, it is not fully liquid, it is semi-fluid matrix present between cell membrane and nuclear membrane. Okay, so nuclear membrane is nothing but the cell wall. It is like a cell wall of nucleus. Okay, that is also used to protect your cell nucleus. Okay, so the thing which fills between cell membrane and nuclear membrane is cytoplasm. எப்படி சொல்லலாம்னா it is like water filled in the bucket. Okay, so bucket is like any container. Say I'm saying is a bucket. So bucket is like your cell wall. Outer layer of the bucket is cell wall, and inner layer we have cell membrane, and the liquid which we filled inside it is called cytoplasm. Okay, so inside that we are going to put some mug and all, right? That mug acts as a your nucleus. Imagine that mug as a nucleus, and the outer cover of the mug is called your nuclear membrane. Okay, like that. Just imagine. Okay, so it is cytoplasm is nothing but semi-fluid matrix which is present between cell membrane and nuclear membrane. It has various cells inclusions called cell organelles and non-living substances called ergastic substances. Okay, it is nothing but the thing which supports make active all the components, functions, units of cell, right? So this is protoplasm and cytoplasm. And next comes nucleus. This is very important. Nucleus. Okay, largest cell organelle present in eukaryotic cells. It is usually spherical. It is like a round shape. Okay, spherical in shape and a transparent granular matrix called nucleoplasm. Inside the nucleus, we are having a transparent material which is said to be a nucleoplasm. Okay, so chromatin network composed of DNA and histone proteins. Inside your nucleus, you will have DNA, deoxyribonucleic acid, and histone proteins. Okay, all these cell functions are storing the protein and convert those nutrients into energy. Okay, and next it is of a spherical body called nucleolus. Okay, nucleus is important, which is present in the center of the cell. It is of spherical in shape. It has a granular material inside your nucleus is said to be nucleoplasm, and it is also composed of deoxyribonucleic acid and histone proteins. The function of the nucleus is to control the center of the cell. Okay, it is the control body of particular cell, and it contains genetic material, DNA. Okay, so DNA which regulates metabolic activities. Metabolic activities அப்படின்னா your intake of food is converted into some energy, right? It is taken done by your digestive process and all. Okay, mixing of oxygen and digestion takes place with the help of this nucleus. Okay, that is said to be metabolic activities. Proper digestion தான் metabolic னு சொல்லுவோம். Intake எடுக்கிறோம், proper digestion ஆகுது, excrete ஆகுது, waste excrete ஆகுது, digest ஆன foodல இருந்து nutritions absorb பண்றோம், equally split to the body, given to another organ, every organ of internal organ of human body. Okay, every organ have individual cells. Okay, that absorbs all the nutrients present in the human body, right? So DNA material is present in your nucleus. So DNA is nothing but genetic material. Next, chromosomes. Chromosomes, nucleus of an non-dividing cell has network of fiber called chromatin. Okay, so it will not divide. It is present in your cell. It is like a fibers. Okay, network of fibers is called chromatin. Okay, nucleus of non-dividing cell has network of fibers called chromatin. Inside your chromosomes, we have chromatin. Okay, and we have chromosome. This is also a transmission of characters or genes. It is used to transmit the signal or the thing proteins from one function to another function, and the chromosome has centromere at the center and arms on either side called chromatids. Okay, இது வந்து ரெண்டு பக்கம் arms இருக்கிற மாதிரி இருக்கும் சரியா, that is called chromatids. Okay, the chromatic thread like chromonema. When you look at the diagram, you could easily understand what is exactly the chromosomes are. Okay, so inside the chromosomes, you will have chromatin and chromonema and chromatids. Okay, chromatids is nothing but the centromere. It is like arms of your chromosomes is called chromatids, and non-dividing cell is called chromatin, and the chromatid thread-like structure is called chromonema. Membrane bound cell organelles. Okay, endoplasmic reticulum. So this is endoplasmic reticulum is a tubular structure. It is also used to extend the cell membrane to nuclear membrane. The flattened sac-like structure is called cisterine. Okay, there are two types of endoplasmic reticulum. One is rough and the one is smooth. Okay, so this is used to support the function. Okay, it supports the function. Actually, it supports the function endoplasmic reticulum. Okay, the function of the endoplasmic reticulum, it mechanical support. I told you, right? It supports the function mechanically, and it detoxification of metabolic waste. It detoxify the metabolic waste. Okay, detoxify in the sense the toxic materials will be eradicated with the help of this endoplasmic reticulum. Okay, and next comes Golgi bodies. Golgi bodies or Golgi complex or same. Okay, so what is the application of this Golgi complex? So we have few more things to follow. Rest of the thing we'll see in the next class. Okay, Golgi bodies, mitochondria, everything we'll see in the next class. Okay, if you have any doubt, let me know. Actually, it's time's up. If you have any doubt, let me know. Any doubt so far? So today's class is fully deals about the basic unit of cell. Okay, basic unit of human body is cell. How the cell is, how we are going to study the cell, how classify the cell is into eukaryote, prokaryote, and size and shape. Everything we have studied in this today's session, and the functions of each and every individual things present in the cell, like cytoplasm, protoplasm, endoplasmic reticulum, chromosomes, chromatids. Okay, everything we have seen today, nucleolus, nucleus, rest of the thing we'll see in the forthcoming classes. Okay, it is very easy subject. It is it is like interesting one. If you are really interested, it is very interesting. Okay, this is very helpful in real life. Okay, so if you're going to hospital, the reports and the scanning things, how to view the scan, okay, how to analyze the uh, things, everything will be studied in this medical electronics course. Okay, so thank you. If you have no doubts, we'll wind up. Any response? Yes, thank you. You can leave the class. Thank you all. Thank you all. Signing off. Thank you.